Hello friends, in this video we'll talk about chromatin immunoprecipitation assay. The protein-DNA interactions play a crucial role in several cellular functions including gene transcription, DNA replication, DNA repair, cell cycles, etc. The CHIP assay is one of the powerful techniques to examine such interactions in intact cells. The gene expression pattern of cancerous cells and any cells under stress differ significantly from their normal counterparts. The transcriptional regulatory mechanisms and chromatin modifications define such differential gene expressions. To have clear understanding of these processes, we should study the binding of regulatory molecules to specific promoters and histone modifications in vivo. These crucial events are based on dynamic protein-DNA interactions, and despite their dynamic nature, the processes can be freezed chemically or probed by immunoprecipitation of protein-DNA complexes. The CHIP assay can therefore be also used in analyzing the binding of transcription factors, DNA replication factors, and repair proteins. So let's see how CHIP assay works. The intention is to examine the interactions of protein DNA occurring in cell nucleus. The assay begins with protein DNA cross-linking by the use of fixatives like formaldehyde. There are histone proteins and non-histone proteins. The histone proteins here forms the nucleosomes. The use of fixatives like formaldehyde fixes or preserve these intact proteins at their specific DNA binding sites. And for efficient immunoprecipitation, the protein DNA cross linkages are processed for the fragmentation into smaller workable fragments, usually of 200 to 1000 base pair, and it is done either mechanically by sonication or enzymatically by the use of micrococcal nucleases. Then for the immunoprecipitation, the antibodies associated with beads are designed against the protein of interest. Here the protein of interest may be transcription factors, modified histones, co-activators, repressors, etc. And the use of these antibodies pull down or precipitates these protein of interest along with their associated DNA sequences. The immunoprecipitated samples are then processed for cross-link reversal, usually by extended incubation at 60 to 65 degrees centigrade, where the associated proteins and antibodies are released from the DNA sequences. Then after DNA purification, the enhancement of certain DNA sequences can be analyzed either by quantitative real-time PCR or microarray or DNA sequencing. So let's see how formaldehyde mediates protein DNA cross-linking. Firstly, these formaldehydes are proper choice for the cross-linking. It is because these aldehydes are highly soluble in water and they are active over a wide range of buffer conditions and temperature, plus they are easily permeable from the biological membranes. That's why they can facilitate the protein DNA cross-linking in the intact cells. These smallest aldehydes, these are electrophilic species. That means they are highly susceptible for the chemical attack by the nucleophilic species. Here the nucleophiles may be from the amino acids of the proteins. Usually formaldehyde targets the primary amino group of the lysine residues. So the covalent bonding forms the addition product methyl oil, which will release the water molecule and form Schiff's base. These Schiff's base are highly unstable. They either decompose readily or they can further be attacked by another nucleophilic species, here preferably from the amino group of the DNA base. That's how it forms the cross-linking between protein and DNA. So here sometimes the nucleophilic species to react with formaldehyde may come from another molecule called quenchers. These are also easily permeable through the biological membranes. These are small molecules like glycine or trace. So they also act in similar way as a protein with a formaldehyde and forming the cross-link product of quenchers and DNA products. That's how it quenches or stops the formation of protein DNA cross-linking, and it is used as the sink for the unreacted formaldehydes. So as I said, the quenchers may be glycine or trace, and it works in a similar way as the protein reacts with the formaldehyde. It also forms Gibbs base, and the quenching products is formed between the quenchers and DNA. So there is cross-linking between quenchers and the DNA. The main intention of shearing or digestion of protein DNA cross-linkages is to fragment them into smaller workable pieces, usually of 200 to 1000 base pair. It enhances the chromatin accessibility to the antibody reagents, and whether to do it by mechanically or enzymatically depends upon the expected chip process to use. For example, in the enzymatic digestion, the endonucleases, micrococal nucleases are used, and it is mainly favorable for N-chip method, that is native chip. These endonucleases or micrococal nucleases clips in between the nucleosomal portions. That's how it generates the monosomes of nearly 175 base pair. So it's used, it's favorable for analyzing the histone modifiers. And these nucleases favors some portion of DNA, some portion of genome to the others. That's how it may sometimes be biased and doesn't produce the evenly fragmented portions. For consistency in such shearing or digestion, we should go for mechanical digestion. So it is done by the sonication, where the high frequency ultrasound waves are used, which will create the randomly sized DNA fragments of nearly 500 to 700 base pairs, and it is majorly favorable for X-chip, that is cross-linked chip method, where the starting material may be the cross-linked chromatins, and these ultrasound waves have access to the formaldehyde cross-linkages, where normally the endonucleases are restricted. When enrichment of certain DNA sequences after immunoprecipitation is done by quantitative real-time PCR, it is called chip qPCR. It can be done either in 96 well plate or 384 well plates in minimal reaction volume. The qPCR primer synthesis can also be done in minimal cost, so it's cost-effective way of analysis. When genomic binding sites are known, the qPCR primers can be designed accordingly, and the amplification of those genomic binding sites can be evaluated. But when genomic binding sites are not known but candidate target genes are available, the primers can be designed against the promoters or conserved non-coding sequences, and the amplification of those primers and non-coding sequences infers the target genes. But when both candidate target genes as well as genomic binding sites are not available, the analysis can be done by microarray or DNA sequencing. So when chip analysis is coupled with DNA microarray, it is called chip-chip method. Here protein DNA interactions can be analyzed in genome-wide manner. It consists of gene chip or DNA chip, which contains PCR products or oligonucleotide probes that represents the genomic sequences. And here the hybridization intensity of chip DNA to the input DNA, it infers the genomic sites interacting with certain proteins. When chromatin immunoprecipitation results are analyzed by DNA sequencing method, it is called chip-seq. Here the chip results are analyzed by next generation sequencing, which provides the genome-wide look at all the locations throughout the genome bound by the protein. The chip recovered DNA are processed for addition of sequencing adapters at both ends. It is followed by DNA sequencing, and after sequencing it is aligned with the reference genome or transcriptome, and based on the alignment scores the genomic browser track are made, which gives us the idea of all the locations throughout the genome bound by the proteins. So it infers the identification of genomic locations of the protein interactions. So conclusively, what we can say is chromatin immunoprecipitation assay can have access to dynamic protein DNA interactions occurring in vivo in chromatin environment, and it can be used for analysis of regulatory molecules binding to specific promoters and alterations in histone modifications, which will be helpful for studying the differential gene expressions in various conditions. So I hope it will be helpful for you friends. Thank you for going through this video.